So my name is Sandén. I'm a researcher from Lund University in Sweden, and these are my disclosures. I am a co-founder and the chief scientific officer at Lead Biologics, which is the spin-out company from Lund University that develops these SLAMF6 blocking antibodies, including TNC1, that we present here today. So immune checkpoint inhibitors have truly revolutionized cancer treatment over the last 2 decades. Most notably, of course, PD-1, PD-L1 inhibitors in many types of solid tumors. But these treatments have been surprisingly ineffective in leukemias, including acute myeloid leukemia, AML. And one reason for this might be that these leukemia cells harbor additional protective mechanisms that shield them from the anti-leukemia T cell and immune cell response. So what we did in this study was that we identified one such mechanism that AML cells use to protect themselves from the immune system. And we showed that if we can block this mechanism, we can once again enable T cells to kill the leukemia cells. This study started with a surface marker screen where we looked at proteins expressed specifically on leukemia stem cells, but absent from healthy normal hematopoietic stem and pre-entry cells. And we looked at patients with TP53 mutations, which have a particularly poor prognosis. And then we found this very intriguing marker, SLAMF6, which is normally only expressed on lymphoid cells, but here we found them in a myeloid leukemia, which intrigued us. And when we validated this in a larger cohort of around 50 cases from our own biobank, we saw that SLAMF6 is expressed in 60% of these cases. And I think that's notable because it's not expressed on the cell of origin in this leukemia, so it must be upregulated from scratch in a majority of cases, which to me suggests that it has an important biological role. So we did a lot of functional work to try and figure out that role using CRISPR-Cas9 and blocking antibodies. And what we found was that AML cells express SLAMF6, and this binds to SLAMF6 on T cells, and this inhibits T-cell-mediated killing of the cell, so it protects the AML cell. And if we knock it out, or if we block it with an antibody, we can once again enable the T cells to attack and eliminate the leukemia cells. And we showed this in multiple models in vitro and also in humanized mice in vivo. Yes, so a key part of the study, of course, is the TNC1 antibody. We developed this in collaboration with the SciLife Lab drug discovery and development platform in Sweden. It's a national infrastructure. So we used phage-display technology to generate an antibody directed against the dimerization site on SLAMF6, which is the site that mediates this protective effect. And we show this is data from co-cultures in vitro, where we mix immune cells and leukemia cells and allow the immune cells to attack the leukemia cells for 3 days in a dish. And we then see that when we add this antibody, you can see you get a very strong dose-dependent effect in inducing immune cell-mediated killing of these leukemia cells. And this works in all the cell line models that we tested. And I should also note that these are all models where the current generation of checkpoint inhibitors against PD-1, PD-L1 are not capable of inducing T-cell-mediated killing. So it seems that this is a very potent mechanism that we have identified. Yes, and finally, we also tested proof of concept in humanized mice. So these are NCS mice reconstituted with human PBMC, so we get human immune cells in there and also human leukemia cells. This gives a very short treatment window of only 2 weeks, but despite that, we still get a sizable reduction in the number of leukemia cells, both in the bone marrow and in the spleen. And we can also reduce the beginning size of these enlarged spleens that are caused by the leukemia progression in the mouse. So to conclude, SLAMF6 upregulation is a novel and very common immune escape mechanism in AML. SLAMF6 serves to protect the leukemia cells from elimination by the immune system. And we have developed this antibody called TNC1, which blocks this protective mechanism and enables the T cells to once again kill the leukemia cells. And targeting SLAMF6 is a new way of addressing immunotherapy in AML, and we hope and believe that this new generation of checkpoint inhibitors will eventually be able to enable immunotherapy in AML and hopefully cure these patients who today have a very poor prognosis.